I'd like to welcome you. My name is Jonathan Friedman. I'm the Development Director here at the Parkinson Institute, and it is our pleasure to co-host the second Stem Cell Awareness Day celebration here. We did this last year, and we're doing it this year. And I've got a few remarks, which I'll be very brief about. First of all, science, in particular stem cell research, has blossomed or possibly even exploded over the past years. Through the California Institute of Regenerative Medicine, the pursuit of this research has grown tremendously, as you all know. You could compare stem cell research with Facebook, as a matter of fact. I recently saw the movie Social Networking. 5 years ago or so, no one had even ever heard of Facebook, and now it's part of almost everyone's daily life. Likewise, stem cell research was relatively unknown, but maybe back about 10 years or so. Now, research is being pursued by hundreds of organizations, universities, government agencies, private industry, and institutes like the Parkinson Institute. The reason is that it holds a bright promise for the future to cure or alleviate many otherwise incurable diseases. So we are here to celebrate the accomplishments that have been made thus far and the incredible hope for the future. First, some basics about today. You've each received a packet of information. It includes an agenda, information about the Parkinson's Institute, information about San Jose State, CIRM, and some facts about Parkinson's disease. Please take the time to read over the information. I won't be reading the speaker bios, but they're all very impressive. Next, the restroom facilities are located behind me to my left. You are right, over here. Refreshments are available at the table over there. Those are provided by CIRM, California Institute for Regenerative Medicine. We will have 4 speakers, then a period for questions and answers for the panel. We'll distribute cards to fill out for those of you who have questions, and we'll select the questions and distribute them to the panelists. So, our first speaker is Christopher Thomas Scott, who is the director of the Stanford Program on Stem Cells in Society. His background is unique in that he blends academic, administrative, corporate, and research with particular emphasis on stem cells, just the right mix to address the ethical and legislative issues surrounding stem cells. So please join me in welcoming Christopher Thomas Scott.
I'm going to ask you a question before we start. How many people here are from Ohlone College? Okay, so you're all on that side of the room. How about, is there a group of high school students here? Oh, you're all over the place. Okay. All right, so I teach several classes at Stanford. I always like to start off with a question, and so I'll start off with a question to the audience. And just signal your answer with a show of hands. So when we talk about embryonic stem cell research, we often talk about human embryos. And so I'd like to ask a question right off the bat. How many think the human embryo that's involved with human embryonic stem cell research is more than 3 inches big? So that's about this big. So how many thinks it's bigger than that? Okay, 2 inches. 1 inch? 1. Okay, how about a quarter of an inch? Okay, we got 2. Smaller than a quarter of an inch? All right. You think it's bigger than a period at the end of a sentence? Yes? Okay, that's good. So this blue thing, which is computer rendered down in the right-hand side of this first slide, is a graphic representation of a human blastocyst. That's the human embryo at 2 to 4 days. That's, of course, not to scale. The right scale is about 0.02 millimeters across, which is about the size of a full stop at the end of a sentence. So when I'm going to give you the introduction to the biology of embryonic stem cells and a little bit about other kinds of stem cells. When we talk about embryogenesis and the embryo that's used to obtain human embryonic stem cells, we're talking about something that's about that big. You can't see it with your bare eye, not usually if you're looking at a dish of cells. So looking out in the audience here, I see a lot of folks with different ages and different experiences and at the different times of life. And the interesting thing about us is that we are falling apart every minute. And as we get a little bit older, we fall apart more faster. I do, anyway. We also get built up just as fast. And that mechanism for breaking down and rebuilding is really at the heart of regenerative cells and stem cells in particular. So just to give you an idea of which systems in the body are on overdrive for replenishing cells, I've given you 3. If you didn't have a replacement stem cell in your gut, you'd lose your intestine in about 2 days, the inner lining specifically. Skin would fall off your bones in about 3 weeks. Your red blood would dry up to dust in about 4 months. Without stem cells and regenerative powers of stem cells, we would have a really hard time living. Where this all really begins is the human embryo. And the human embryo at 2 days, that blue thing I showed you, is now here shown in cross-section. And if you want to see a little bit more about this, you can buy a book that I wrote called Stem Cell Now. It has a good introduction of stem cell biology in it. It's on Amazon, and you can pick it up for about 15 cents. But if you take the embryo and you slice it in half, you see a little mound of cells along one margin, and that is called the inner cell mass. So if you go to a cocktail party with a bunch of stem cell biologists, you can say, hey, tell me about the inner cell mass, and you'll really impress them. But the big experiment was done in 1998 when a guy at Wisconsin, University of Wisconsin, his name is Jamie Thompson, found that if he could take that inner cell mass out of an embryo in a process that destroys the embryo, takes those cells and he puts them on a petri dish with some other ingredients, including feeder layer cells, and lo and behold, he gets embryonic stem cells. The principal features of embryonic stem cells is that they are, at least as far as we know, immortal. They're genetically stable, and they have a feature called pluripotence, which means that they can theoretically develop into any of the cells and tissues of the body. And it's that last attribute that makes them so darned exciting. This is embryonic stem cells that I made. Yes, even I can make them at Stanford using James Thompson's first line. So these cells are from that original line that he discovered in 1998. And the cells, the embryonic stem cells, are in the middle. Those kind of skinny-looking cells on the outside are the cells that feed them and keep them alive. And these things will just go forever. The important, I guess, part of this is to understand how embryonic stem cells really come to be. And an interesting kind of footnote to all of this is that embryonic stem cells is really a term of art. So embryonic stem cells really don't exist in the organism. They exist on the laboratory bench. And here's a picture, a real picture now, of embryogenesis from day 1 to about day 4. These pictures are not to scale, by the way. But the fertilized egg is over on the left, and then here's a photomicrograph of a human embryo at about day 3 or 4. You can see the inner cell mass kind of nestled around one side there. I mentioned that embryonic stem cells are pluripotent. That means that they can change into any of the tissues, cells in the body. What does that mean in terms of their power for medicine? Well, this is kind of a schematic here. So the first thing to notice here is that this process of forming an inner cell mass starts with fertilization, then moves to the blastocyst, as I've shown you, at about day 2 or 4. And then I've shown you a little bit about how that inner cell mass then becomes a line of embryonic stem cells. You often hear in the news, lines of embryonic stem cells are used. This is what that means, that culture. And then the theoretical next step is that these cells can make different cell types in the body. And scientists have shown that this is indeed true in many, many cases. And every week, it seems, they're finding more and more different kinds of cells that the embryonic stem cells can actually make. And then the end product of this, at least from a therapeutic point of view, is that these cells could then be transplanted into someone who needs them. So potential cures, treatments for a variety of diseases is what's driving, I would say, most of the research right now using embryonic stem cells. Because these cells come from a 2-day or 4-day human embryo, however, they come along with a lot of controversy, and I'll mention some of the legal and political ramifications of that at the end of the talk. That's one technology. At the same time, scientists have they're cooking up other ways of making these powerful kinds of cells. And one way they do that is something called direct reprogramming. And reprogramming cells is a technology that came onto the scene in about 2007. And it looks like, at least, that these cells have many of the properties of embryonic stem cells. We don't know right now if they are going to be as powerful as embryonic stem cells. And so it's very important that we keep both of these lines of research going at the same time, because we want embryonic stem cells to be compared with other things that we discover. But to just describe briefly how this works is that scientists are able to take a cell from, let's say, an affected person, like a patient, and inject into that cell 4 genes. There have been experiments now with less than 4 genes and some with no genes at all. But the basic experiment is to put 4 embryonic-like genes into the cell. They inject it in with a virus. Viruses do this kind of naturally. These cells then are reprogrammed, and it's just like computing. You reboot the cell to its embryonic state, and then the cell goes on and becomes very much like embryonic stem cells, pluripotent. So the advantage to this versus the technique I just showed you in the slide prior is what? Can anyone identify it for me? No embryo, that's one advantage. The cells? Yes. So the cells match the tissue of the donor or the person or the source where the cell came from. So this means that you don't have to worry, if this worked, you wouldn't have to worry about tissue rejection. And that is a disadvantage with an embryonic stem cell therapeutic strategy. So this is exciting too. The way you might actually work this in terms of drug development is that you may be able to study, let's say, a disease at a cellular level and then test that disease in the laboratory with certain drugs to be able to figure out which drug would work. Those are pluripotent stem cells. There's another type of stem cell that isn't as powerful, and those are the so-called adult stem cells, sometimes tissue-specific. You can also hear them described as adult stem cells. They actually form during fetal development, so that's not quite a correct way to name them, but it'll work for us. And these stem cells are maintained in our tissues. So when I opened up by saying we're all falling apart, well, it's because of these stem cells that we stay together. And they hang out in various tissues and organs and replenish cells that are either injured or are dying. And we know of adult stem cell populations in dozens of areas of the body. Important ones would be the blood, brain, gut, as I mentioned, skin, hair, lung, heart, and other places. So there's a lot of ambitious research going on in each of these areas for the various things that they could be applied to. Moving from kind of the theoretical to the actual, this is a a basic rundown of where stem cells are now in terms of their application for humans. These are 7 or 8 companies that have been taking some of these discoveries and moving them into the clinic. They range anywhere from macular degeneration to Batten disease. This is a company that's right here in the Bay Area. And then most of you may have heard of the Geron trial that was just recently approved by the FDA. That trial has also just recently been approved at Stanford, so it looks like that we'll be doing a part of this study at Stanford University. And this one is for spinal repair using a type of cell called an oligodendrocyte. So the idea behind most of these studies is to see if these cells are actually safe. So the first idea here is to put them into a small group of patients and see if they are safe. And once we move past the safety part of the study, we move into dosaging and the rest to see if they actually can produce some sort of clinical impact in patients. So this is all very exciting. One of the things my group at Stanford studies is this transition between what we say we can do in the clinic and what other groups say they can do without good scientific evidence. An alarming thing for me as someone who works in ethics is the rise of something called medical tourism. So this is where patients, people, their families travel to other countries to get therapies, and I use the term loosely. Usually they are just injections of cells to treat their particular disease. And the reason it's alarming to me is because there's very little scientific proof that it actually works. One of the things we do at Stanford is we track these so-called clinics, which countries they're in, how they're recruiting patients, what the costs are, and we regularly publish results on how offshore tourism is actually developing using stem cell treatments. And I just draw your attention to the fact that this isn't something that's just isolated to a single country. It's all over. China, Mexico, certainly, and actually here in the United States, there have been a number of cases where there are clinics doing unethical experimentation on humans. In terms of how many people that are being put through these clinics, it's really hard to know. And this is an area of very active study. I think it's probably in the tens of thousands, maybe even more. And one particular company in China called Biomedical Ethics has run through maybe thousands of patients through its clinics using a type of cell called an umbilical cord stem cell. And their claim is that umbilical cord stem cells can cure maybe 30 or 40 different diseases. It's just simply not true. Cord blood cells do a very good job in certain cases in cancer, but it's not clear that they will do the trick for more complex diseases such as Parkinson's disease. But I mentioned Belky here because I have a student who actually went to a Belky clinic. And I didn't know that he had gone until after he came back. And I asked him to tell the class about his experience, and he did. And so he gives lectures in my stem cell class every quarter on his progress since he's come back from Belky Biotech. He has something called Friedreich's ataxia. It's a severe degenerative neural disease. His father had it. He's, I think, in his 30s. He came back to Stanford after a successful career in engineering. He wants to be a biologist now. And he did a very interesting thing before he went to China. He goes, you know, I know that this is pretty much an untested procedure. So what I'm going to do is I'm going to go to my doctor, a neurologist, and I'm going to have her check me out before I go and then after I come back. Now, for those of us that are in the sciences, we know that that's a very small number to base a result on. But it's interesting, nonetheless, that he would think about this. And so the reason I ask him back every quarter is that he's, the scientist in him, is looking at his own progress, and he's now reporting it to the class. And long story short, Richard is about a year and a half out of his treatment, and his progress at the beginning was interesting. He actually improved the first 3 months after he came back. So he could write much better. He could stand without assistance. He could talk without slurring. So this was looked like an interesting result. However, since that time, he's now below baseline. So he's about where he started, maybe a little below. So the reason I think that this is worth telling you about is that we need, as scientists and as researchers, more data about what these effects are for people. And also we need to be very clear about what we believe the safety and the risks are for people going to clinics. Richard had no clue about the risks. He got 4 lumbar punctures of these cells while he was there. And with some clinics, at least we know that's a recipe for disaster, specifically spinal meningitis. So this is a really interesting case. If you're interested in reading the case of Richard Chin, it's in the American Journal of Bioethics. We just published this about 2 months ago. He's also been featured in a number of news programs and another article in the journal Nature Biotechnology. So I'll end with a couple of slides here that I think are kind of interesting. These are old slides from about 100 years ago. And I'll bring you kind of through them one by one. This is the one I like the best, actually. This is an electric belt for weak men. And it's for guys who are debilitated, seminal weakness, they're impotent, they've lost their manhood, they're nervous, they're sleepless, and just in case they got something else wrong, it's for general ill health. And then here's another one from about the same period. This guy, his name is Dr. Johnson Can Cure Cancer. All you have to do is read his books and pay for the books. And then, of course, you can wash away years of fat. And then here's one that interested me, specifically Hope for the Bald. And this guy is Dr. O.E. Horn. And apparently Dr. Johnson never talked with O.E. Horn because he's bald. Look at the language here. There's a whole book of testimonials from people that he's supposedly helped. Now, let's fast forward. This is a clinic in Geneva. We're going to give you stem cells if you have a general reduction in vitality, if you're depressed, if you have arthritis, endocrine disorders, sexual impotency, chronic pains, sequels of illness. No idea what a sequel of illness is. So you can see that for a fair amount of cash, you can go to a clinic in Switzerland and have a cure for what ails you. Here's perhaps the biggest website for trafficking in stem cell clinics in the world, and it's called China Stem Cell News. It looks like a news portal. It is not. It's actually a site that's run by a company, and this company has testimonials, just like those old ads I showed you on this side and this side. And if you go into the site, it will have a pull-down menu so that if you have a problem or a disease, you can pull down your disease, and then it will give you a registration form, and they will find a clinic for you. This is where Richard, my student, found his clinic. And the reason I use this as an example is that the internet is really the tool of choice now for putting desperate patients in touch with those clinics that may be putting stem cells into them in a risky or unethical manner. Last couple of slides is just the situation here in the U.S. Another research project we do at Stanford is we track and publish on the various interesting things about state law. We're lucky to be here in California, where the California Institute for Regenerative Medicine is moving this research along at a very good clip. Other states are not so fortunate. And in fact, in some states, there are actual measures afoot to discourage embryonic stem cell research. And so with my research group, who includes a bunch of lawyers, I'm not a lawyer, we look at those states that would perhaps restrict embryonic stem cell research in some way. Restrictions could be no funding, it could be no building projects, it could be no tax breaks. It could be a various amount of things. These are the states right now in the U.S. that support it. This is a little old, so this could be a little bit different. And the interesting thing is this group of what I will call the states with no clear position. These states have not decided which side they're going to fall on. I usually show this to students who are considering careers in stem cell biology because I think that being an informed citizen about the laws, about where you're going to go, if you want to make your career in Kansas, you should be pretty careful, I think, because Kansas at least has no stated position on it, and if the legislatures decide next year that they want to put someone in power that would, say, make it illegal, your career might be at risk. The sad thing about this as a policy researcher is that we have now a very fractured way of doing research, especially embryonic stem cell research in the United States. And now with the recent court injunction, we've got an even bigger problem because now all embryonic stem cell research is in jeopardy. George Bush in 2001 had his criteria for federal funding. It limited research to a certain number of lines. Our group was the first to publish a paper that actually identified how many lines had been available over the course of his 8-year term, 18. 3 of them are used most often. But these were the basic rules that President Bush set up. And many of these are with good ethical grounding. For example, that the donors have granted their informed consent to use the embryos. This is all good things. The thing that made things tough on scientists was that there just wasn't much material to work with. So during the course of Bush's years, the Congress introduced, and in some cases passed, depending on which house you were in, a number of bills that would actually make embryonic stem cell research illegal. So now I'm not just talking about you can't do it because you don't have any money. Now the question is, if you do it, you go to jail. And it's incredible to me that we had several acts that were actually passed in the 109th Congress, 108th, 109th Congress, that at least in the House, that would have criminalized embryonic stem cell research. Luckily, none of them went through. In fact, the 2 bills that did go through was a bill that would actually loosen up the restrictions on embryonic stem cell research and provide funding. It's called the Stem Cell Research Enhancement Act. It was first introduced in 2005, I think, reintroduced in years past. Both the House and Senate voted to approve it, and President Bush vetoed it. Not once, but twice. So that's where we were when Barack Obama came on the scene. He began a new discussion about embryonic stem cell research, and within a short period of time issued his executive order that basically revoked the Bush policy. And he instructed the federal government to support the research, to specifically fund research. So this was the will of both President Obama speaking, and now I've shown you in the slide prior that we had the will of Congress that was also speaking. But in August, embryonic stem cell research, at least for that period, was stopped in its tracks by something called the Dickey-Wicker Amendment. This is actually a rider on an NIH bill that has been around since 1995 in one form or another. And basically it says that researchers may not use federal funding on any research in which a human embryo or embryos are destroyed, discarded, or knowingly subjected to risk of injury or death. I go back to the first slide. This is the human embryo at 2 days we're talking about, including those embryos that are regularly thrown away in vitro fertilization clinics. In August 23rd, we have a district court judge that interprets all embryonic stem cell research to be covered under the Dickey-Wicker ban. And as a result, not only can we not use embryos, but we cannot do embryonic stem cell research. This particular ban, the injunction, has been stayed temporarily while the courts figure out a way through it. So we're in this limbo right now where we're not sure what the arguments are going to be that determine the future of embryonic stem cell research. Lawyers and scholars that I talk to think that it's going to be overturned and we're going to get on with business as usual. If you want a prediction, I think that what we're going to see in the next few months and perhaps year or so is that bill that I mentioned, the Stem Cell Enhancement Act. It will be reintroduced, and it will be introduced with specific language that will take care of the problem with Dickey Wicker. And that language may be something like researchers might be able to use embryos that are donated from in vitro fertilization clinics for research. That would be a compromise wording that you might expect in a legislation like this. So we'll see. And with that, I'll end and take a few questions. Where did the Dickey-Wicker Amendment come from? Or where did Dickey and Wicker come from? You know, I don't know which state they come. That's a very good question. My guess would be probably conservative states. Anyone know? Reach back that far into your memory? So the question is, are there barriers to using, let's say, mouse embryonic stem cells, and are there, do mouse embryonic stem cells represent a good model system for using human? The answer to the first question is no. Animal embryonic stem cells can be used without any problem, and the mouse is the best model. Does a mouse embryonic stem cell tell us everything about human embryonic stem cells? No. And for obvious reasons, mice are much different than people. And we need the two actually running closely in parallel so we can see what the differences are and do the experiments that would move one along. In fact, James Thompson, the reason he was such a genius is that he got a mouse embryonic stem cell procedure to work using human lines. It took him many years to do that. The question is kind of a follow-up on this one, and would a different species, let's say more related to man or human, such as a chimpanzee? Yes. The primates are a place where a lot of this is being studied. Primate embryonic stem cell lines are difficult to make. There are a few of them that I know about that are in culture. And the larger animals in general are a little more difficult to work with. They're large, for one thing. They're expensive, for another. And the so-called species barrier jump from, let's say, a marmoset, which is a small primate, to a larger primate, such as a chimpanzee, sometimes is all it takes to frustrate years of research. So not even within a group like primates will you get the same sorts of results. That's a good question. Right, so the question is about umbilical cord blood cells and matching. Yes, umbilical cord blood cells have been shown to be very effective in certain types of childhood cancer and in certain adult cancer diseases, lymphomas and others. The matches for transplants are very much like the matches for other kinds of transplants where you're looking for close relatives and siblings. So a SIB transplant from one unaffected child to one that's affected works very well. In fact, some cancers have actually been cured that way. Whether or not umbilical cord blood cells will cure Lyme disease, we simply do not know. And that's where some of the claims get very, very dicey. Thanks to Google. Dickey is from Arkansas. Wicker is from where? Mississippi. Another weakness, I guess, in some particular diseases is that if you're taking a disease cell out and then trying to fix it and putting it back in, you have to make sure that it's fixed. Otherwise, you're just reintroducing the disease back into the person. So with induced pluripotent cells, I think this is a big concern. Not only do you want them to be their embryonic state, but you want those embryonic lines to be free of perhaps the genetic disease that is ailing that individual. Yeah, great question.
Dr. Tzvia Abramson is Associate Professor in the Department of Biological Sciences at San Jose State University. we collaborate closely with her in our own stem cell research, and through the university we've been fortunate to receive several interns, one of whom we have recently engaged in a full-time position here. For the students who are visiting from some of the schools in the area, there's tremendous opportunities for you at universities and with research institutes like ours. So I encourage you, if you're interested in science and in stem cell research in particular, stay tuned because there's a lot of things coming our way. So without further ado, I'd like to introduce Dr. Tzvia Abramson.
I would like to just mention the program that's going on at San Jose State, and also how lucky we are that we are actually in one of those green states that Chris mentioned before. Because of that, we get to play and investigate and research stem cell more. And that's due to a very important company in California, California Institute for Regenerative Medicine, which in 2004, because of Proposition 71, as many of you probably know, they got approval to fund stem cell research in California and will issue a grant of $3 billion that can be distributed to researchers and state university like us and community colleges, so we can boost this very, very important work in California. And we are very happy to be able to participate in that. San Jose State University, among other 16 state university and community colleges in California, 3 years ago, obtained quite a lot of money to put programs together to boost the workforce in stem cell in California. And our university obtained $1.73 million to put such a program together. And different universities do it in different ways. At San Jose State, this is a program that we issued for graduate students, for master's students, which are screened pretty intensively for this program. And it's a 2-year program where the first year they take classes at San Jose State, and second year they get to go to an internship, a full-time internship. They go to a 1-year internship in a couple of laboratories in the area that host our students, and Parkinson Institute is one of them. But also Stanford University, we have 62 scientists at Stanford that host our students. Santa Cruz University, UC Santa Cruz hosts us, and the biotech company Escape Therapeutics in the area also have a couple of our students. So they take classes related to stem cell for the first year of their internship, and then second year they go to this place for a full-time internship. We also, because of this grant, we actually started this semester. This semester is the first time that we are giving away this class, a stem cell class, a full semester stem cell class, which is primarily for the students in this program, but also for the rest of the students at San Jose State. They go over the curriculum of stem cell, how to handle stem cell, how to manipulate, how to work with them in the lab, and how to differentiate those stem cell into different lineages, such that when they get into the workforce, when they are hired by the company, it's a much smoother transition, and they don't have to go for many months through this program, through this practice, so they right away are efficient in their jobs. So it's a whole program that we actually put together as this little poster explain, and most of our faculty is enrolled in that. So each one of the students that's enrolled in the program has a San Jose State mentor, a faculty like me and many others, and they have another mentor at the site of the internship. So they are very careful, watched, and followed throughout the program, and see that they are getting the best out of it, and then they are progressing. And a lot of our students are staying, like students in the Parkinson, that they get to be hired right after they finish their internship in the place where they work. But they also are hired in other laboratory and academia and that do stem cell work. So this program is very much valued. Other campuses have similar programs for undergraduates. Our program is for graduate students. Other campuses like Humboldt and other campuses are having a similar program for undergraduates, so they can transition immediately after their BA or BS into those workforce. I anticipate that the progress in California, at least, and again, we are really blessed to be able to work here, is gonna transition really fast. Our students are getting educated in how these stem cells are used, different approaches for the regenerative medicine, implantation, but mainly into issues of model of disease that a lot of the companies are using stem cells. So many of the companies, and in fact in Parkinson, and you'll probably hear more about it, are using stem cell not so much for implantation, although also for that, but really to be able to have a model of disease in a dish, enough cells that companies, biotech companies and the institute can research those cells to identify new drugs, new molecules that can be potential drugs, or identify drugs that can be tested on a disease model in a dish and check their toxicity. So if we have an unlimited amount of cells which are representative of a particular disease in a dish, we can potentially screen for thousands of drugs that are available there and see which one works and which one is not toxic. A lot of the work on stem cell is actually in this direction, in model of disease, rather than implantation or regenerative, which is a little slower because of the problems that Chris mentioned before. It's not so easy to get approval to inject those cells in human. But definitely as model of disease, this is a very promising area where stem cells have a lot to contribute and speed up the way we identify new targets, drug targets, and screen for potential drugs. So we are training our students to enter this force and be competitive and attractive candidates for those companies who are looking for people to boost and speed up the progress of identifying drugs. And it's a marvelous program that students are really interested in to participate in that. We actually have to screen, and because we have a very limited number of places in there. Those classes, though, are going to be offered as part of the curriculum for the regular biology students, so it's not something that's going to be selective in a while, only for students which are enrolled in this program, which is funded by CIRM. So it will be open to all biology students at San Jose State, and they also will have a benefit over other candidates and be able to compete on those jobs that are offered in the stem cell arena in California. Yeah, and I'll take some questions. So each round, so it's a program that has been funded. This is the third year. Each year, it's 10 students that are going through the process. But as I said, we have these classes now as part of our curriculum. So biology student or students from other departments as well, if they have the prerequisites, they can actually register to those stem cell classes. It involves a theoretical part and a very intensive laboratory part for 4 months. Yeah, we already do. In fact, it's part of the grant. We have to do that. We have to follow. So pretty good, pretty great. Yes, yeah. So a lot of them, actually, from the first round, I think 5 of them got to stay in the laboratory where they did their internship. Several of them went to the PhD programs, and we'll see. The second round is about to finish in a couple of months, so we'll have to see what's happening. And we are enrolling more sites, hosting sites, so we'll open more opportunities to students.
Dr. Berta Strulovici, is that right? Close? Is Vice President for Research and Chief Technology Officer at Iperion. Iperion is a pioneering biopharmaceutical company that is taking the cutting-edge technologies of cellular reprogramming and directed differentiation to an entirely new level to harness the power of induced pluripotent stem cells to advance the understanding of human diseases and accelerate the discovery of more effective therapies for patients. I got that from their website, by the way. And they've been backed financially by Kleiner Perkins, Highland Capital Partners, MPM Capital, and Google Ventures, just to name a few, up to about $60 million. So it shows the power of stem cell research and what they're doing and how it's attracted investment, particularly in this area. So please help me welcome Dr. Berta Strulovici.
I'd like to thank Birgitt for inviting me. So my presentation is going to be very, very different from what you heard right now, because we are a company and we need to apply the basic research discoveries to finding a drug. However, I'm going to base a lot of what I'm going to say on what the previous speakers already mentioned. So they described the process of making these cells. They described comparisons between stem cells and iPS cell technology. So I'm not going to go and describe this again. In addition, I'm actually very pleased to hear that we have in the audience a lot of students, because I will consider myself a person who can describe to you how drug discovery is done. And we are always very interested in young talent. I was happy to hear that there are actually educational programs now in stem cells. So we are always on the look for bright, eager students who have some background from their alma mater. So a biotech company cannot operate by itself. In addition to money, it also needs very strong academic advice. We were very fortunate to have several scientific founders and advisors from the Harvard Stem Cell Institute. This is actually very important for us because 99.9% of the research is actually done in academic labs outside of our walls. And we need to make sure we forge interactions with such academic advisors. The only thing I'm going to say here is that in a company, there is leadership organization, which is very similar in every biotech company. You have a CEO, you have somebody heading research, which in this case is myself. You have somebody who deals with the money, a chief financial officer, somebody who deals with business to make sure that our product can be commercialized with other companies. And you always need to make sure you protect your intellectual property by having somebody in legal affairs. So how do we start? Why do we want to work with iPS cells? As it was mentioned before, we actually, our business is not regenerative medicine at this stage. And if you'll ask that question later, I will explain why this technology today is not ready for regenerative medicine, although the idea appears cool. We are using patient-derived cells because we want to look at a disease in a dish. The fact that the power of making these stem cell-like cells and then differentiate them to different types of functional cells that are critical to certain diseases, we start with the presumption that the patient information, that disease information, is actually carried through in these particular cells, and that we will be able to not only study the disease in a dish, but find compounds and other agents that would have an impact in finding potential drugs for the disease. So where are we? We start with patient and end with patients, because we start with patient information and patient material to make our cells. We take these cells and put them in a dish. We use multi-well plates to test for compounds and look for candidate drugs that then we want to put into people in clinical trials. So it's not cells, but it's compounds. So I'll describe just in a few words what it takes to industrialize the production of iPS cells. It's very different from working in an academic setting and when you need to create something on a large scale with certain standards, something that's scalable and reproducible so that every day you get the same result. So what you see here is a schematic diagram of the process that takes us from identifying clinically relevant patients to taking a biopsy, creating fibroblasts or other types of cells, and then using the process that was described before. In our case, we are using retroviruses to introduce 4 key transcription factors that have the ability to reprogram an adult cell to become an iPS cell. Then we differentiate these cells to the type of cells we have an interest in, such as a nerve cell, a pancreatic cell. But then there is a very important step here. We need to assess whether those cells preserve any of the hallmarks of the disease that the initial patient had. So we call this a disease phenotype validation. And with this, we then create something that we call an assay, which is a reaction in a test tube that recapitulates that particular phenotype of disease. And then we use this both to look for compounds that could impact the disease, could serve as the basis for drug discovery, and we also look for novel molecular target identification in these cells to impact drug discovery. What you see here is simply the types of technology and instrumentation that we use in our labs to bring this process from the beginning to the end. And this is why I'm going to say, it was mentioned here that we raised $60 million. It's actually very expensive to do drug discovery. Part of it goes on capital equipment, part of it goes on bringing in talent, scientists that can do the job. So when we are saying putting the patient first, we are referring to the fact this is what you see in this image is a child with spinal muscular atrophy, which is a disease I'm going to describe a little bit to you because it's a proof of concept of how we use this technology. So what does it mean here to put the patient and the disease first? The moment we decide what disease we are going to go into, we are going to look for the type of patients that would benefit from this technology. And we are looking to get all the information about these patients because we are hoping that we will take all their information and be able to recapitulate it in the cells that we took from them. So we go, however, from one individual to create many such lines from many patients in order to create a population of such cells so that not only one person would benefit from the compounds that we find, but many people that have this particular disease. So where do we start? I mentioned earlier on that we take skin biopsies from the respective patients. We create fibroblasts from them. And this is the starting point from which we create our induced pluripotent stem cells. So this schematic diagram just shows you part of what was important to achieve in order to make this process as industrial as possible so we can use it on many patient lines and get the same results over and over again. So today we are using 3 or 4 factors. You heard about feeder conditions that are used with stem cells. We are using feeder-free conditions. And we make sure that our cells are at the earliest passage possible so that they don't acquire themselves mutations and so on, and hope to preserve the phenotype of the disease. Last but not least, you need to know that in a company, you need to be very efficient. You need to be able to have few FTEs do a lot of stuff. So when you think about jobs in industry, you need to know that people work hard, but they work smart. So I'm not going to belabor this, but just to tell you that when you industrialize a process, you need to put in place a lot of quality control measures. And in this case, we needed to put quality control measures that have to do with a stem cell-like cell to make sure that our cells are as stem cell-like as possible, looking for gene expression for pluripotency. We wanted to make sure that genes that are fibroblast specific are down-regulated. We are profiling our cells to make sure that they behave as much as stem cells as possible. We are also making sure that our cells, through the process of becoming stem cells, have still a normal karyotype. We do the STR fingerprinting, which is very important because we need to make sure we never mix up taking the iPS cells from one patient. They have to really come from that patient's fibroblasts, and that we keep that lineage to make sure that the cells are from the patient we think they are from. Needless to say, if you want to do a lot of drug discovery processes with your cells, you also need to scale this to create a lot of cells that you can work with from the same patient cells. This is just a schematic of how this is a normal karyotype. These are some of the genes that appear, that become induced in the process as stemness markers. We also make sure that our cells have the ability to differentiate to various types of cells of interest. So we are looking at differentiation markers for endoderm, mesoderm, and ectoderm. I mentioned until now that most of our work is done with human fibroblasts. We are able, however, to take also peripheral blood and create and reprogram cells to create iPS cells from peripheral blood. This is a publication, is a cell stem cell with one of our colleagues, Stefan Iperion, together with one of our SAB advisor, George Daly from Harvard Stem Cell Institute. And this is actually very good. For example, if we want to create induced pluripotent cells, but we cannot get access to a patient's fibroblasts through a biopsy, we can take blood, and this could be a less invasive process than taking a biopsy. And as I'll move forward and I'll show you our progress in spinal muscular atrophy, this is a disease that affects babies when they are just born. It's kind of difficult to take biopsies from very young patients. Part of having an industrial process means also that you need to be extremely well organized. So we have created a database that contains all the information on the patients that we have biopsies from and all the information on the iPS cell lines that we produce. We also keep track of patient information, actually, because when we start looking for compounds that affect the disease, we need to link back to all the information we have about the particular patients and see if we can actually reproduce any of the disease features in our in vitro experiments and see if the compounds that we identify can affect them. This just shows, again, part of being productive, you need to do a lot. For example, in this case, we have 355 iPS cell lines from skin fibroblasts, and these are the diseases we are working on or plan to work on, and we already have in-house cells from people with ALS, SMA, type 2 diabetes, Parkinson's, and I'll mention at the end something about Parkinson's. So when we are saying we are putting the patient in the beginning, it means that we need to know up front what patient population we are focusing on to find a drug for their disease. So this slide, it's a schematic diagram. Don't worry so much about the wording here. But what it tries to say is the following. When we create iPS cells from one patient line, we have the luxury to differentiate these cells to many cell types. Some of these cells can serve as disease in a dish, and some of those, and also we can produce, for example, liver cells, cardiomyocytes, and we can test from the same patient also for the toxicity of the compound that we find. So this is actually something that the part that has to do with patients is really the huge advantage that induced pluripotent stem cells have over human stem cells, because we are preserving the disease of the patients, and that's something you cannot do with human ES cells, unless they are cells from IVF that were identified with a specific mutation. So I'm going to give you a brief synopsis of what happens in our initial drug discovery program. It'll be an education for people who don't know anything about how you develop drugs in a pharmaceutical company. So this is a spinal muscular atrophy is really a horrible disease. The basis of it is loss of function of motor neurons. Patients lack or lose completely their ability to walk or breathe, and this is actually noticeable in babies at the age of 6 months. And there's absolutely no treatment today for this disease. This is a monogenic disease. It's monogenic recessive, and it is caused by a mutation in one protein, in one gene, that makes the cells produce less of just one protein that is critical to the function of motor neurons that innervate muscle. The incidence is 1 in 6,000 live births. It is a highly solvable disease, but we need to ask ourselves, how come through all the drug discovery companies there is still no drug for this horrible disease? One of the reasons is, of course, that this is what we call an orphan disease. It doesn't have, it's not that, we can't say that 3/4 of the globe in the Western countries have it. Therefore, pharmaceutical companies won't spend a penny on this kind of disease. And the reason I know is because I spent 12 years of my career in such a large company. And also because it's really very difficult to treat very young children. And also because the technologies were not there for people to learn about the disease. Because it affects motor neurons, one couldn't go and take motor neurons from patients and study them. Therefore, with the advent of this technology, now we have the luxury to produce the kind of cells that are critical to the disease, study them in a dish, and actually look for compounds that can treat the disease. So our program goal is to discover and develop a drug that slows or halts disease progression in SMA patients and improves survival and quality of life. That just shows the disease is actually, there is type 1, type 2, type 3, and the severity of the disease depends on the amounts of this particular protein that I mentioned that is reduced in these particular patients. When you do a clinical trial and you want to study the disease, you also need controls. So here we have discordant siblings, carriers, patients that are carriers of the mutation but have not developed the disease. So this is really a miniature clinical trial that we are thinking of doing in vitro. So this is how, for people who don't know, this is how motor neurons look like. We identify them by various markers. Islet in red is typical marker for motor neuron. The API in blue just stains all cells, tells the nuclei of all cells. We are able to create day in and day out motor neurons from our iPS cells from SMA patients and for ALS patients. And that was the first step in being able to develop our drug discovery process. So this is how this disease phenotype looks like. I mentioned that the protein is, there is less of this particular protein, which is called survival of motor neurons. In a control, in a normal motor neuron, as you can see in green, there's a lot of this protein. All motor neurons have it. But in patients, in cells derived from patients with type 1 spinal muscular atrophy, very few of the cells actually express this particular protein. And what we also found is that there are much fewer motor neurons than in control cells. If you want to do a screen using hundreds of thousands of small molecules to see if you can increase the amount of this particular protein in the dish, what you need to have is a very large number of cells, actually billions of cells. So part of what you do in a drug company is you make sure that you can scale your processes. So we are able today to produce billions of cells, and this is a 384-well plate. If you can't see, this is actually a smiley face. The guys in the lab were having fun. If you want to do a 500,000 compound drug screen, where in each well you put a different compound, and you are looking to see in which well there will be a compound that increases the amount of this particular protein in the cell, you need to make sure that you have a really scalable process. And you want to make sure that from the beginning to the screen to the end, you have the same, the cells behave the same way. Otherwise, you can't compare your data from day to day. This is actually what we are looking at. Cells in each one of those is a well. And this is a 384-well plate. Each one, so what you are looking here is the results from a screen where measuring this particular protein in most cases, in most wells, there is no compound that increases the level of the protein. But you can find from time to time, if you are lucky, 1 or 2 compounds that increase a little bit this protein over background. And that's what we are looking for, actually. So when we started this process and wanted to set up a screening assay and look for small molecules, we first went through the literature. We went through reports about clinical trials to see what compounds other people have found by completely different means. And we did what you call a benchmarking. We wanted to see how our assay stacks against what other people have done and try to better understand why compounds that were used in the clinic actually failed. So what we found is that if you take a fibroblast directly and you do a screen, because I forgot to mention that this particular protein is not found only in motor neurons. It's actually a protein that is critical to the function of every cell in the body. It just so happens that motor neurons are more susceptible than the other cells to reduced levels of this protein. So we compared a cell that is irrelevant to disease, which is fibroblasts, and we did the same assay against motor neurons. And we found that out of 27 compounds that have been reported by various groups, including NIH, several companies, some academic labs, including Stanford, we found that although some of these compounds were working in fibroblasts in increasing the levels of this protein, none of these compounds were working in motor neurons. So you can think both ways. You can think that there is something to this, and maybe the compounds have failed in the clinic because they didn't even touch motor neurons, or that maybe our assay is not so good. Going through our first 78,000 compounds, we actually were I started to identify compounds that work only in motor neurons but don't work in fibroblasts, and only one compound that was common to both. So this started to tell us that although this protein is ubiquitous, it may have different roles and different functions in different cells, particularly in the cells that are critical to the disease. And that's something nobody was paying attention when going to look for a compound that to put in the clinic for SMA. So this is a compound. So what you are looking here at this particular graph, just to orient you, we are looking for an increase in this particular protein. So we found compounds, like I showed you before, that work in fibroblasts but don't work in motor neurons, and compounds that don't work in fibroblasts but work in motor neurons. So we decided that most likely these compounds must have different mechanisms of action, and it will actually be interesting to take them forward and see which type of compound would have benefits in the clinic. So this is one compound, which is now our lead compound, that actually works in motor neurons, doesn't work in fibroblasts. And we already identified other types of compounds. We call them analogs, compounds that are in the same family that increase SMN levels only in motor neurons. So with this being said, we are really moving forward with a handful of compounds that work, some in motor neurons and one common to both. And we are going to test them in what we call an in vitro clinical trial, where we are going to take cells from all the patients that we have in-house already, and we are going to see how these compounds behave when you look against a panel of cells from different patients rather than from just one patient. And this will give us an indication of what will work in the clinic way before we actually go to the clinic. And that's a concept that has never been used in drug discovery in a large company. Actually, never in any company. So with this brings us to Parkinson's. We started the program in Parkinson's. It's a lot less evolved. We have very few resources, so we have to be careful how we use them. And right now, a large number of the people are working in SMA. But where are we with Parkinson's? We actually forged the collaboration with with Parkinson's Institute, and that was very beneficial because that was the only way to gain access to patients from familial and sporadic Parkinson's, and also be able to capture the patient information that's absolutely critical. In addition, it's very important to collaborate with the type of people who understand the disease deeply and have direct contact with patients, because they will help us also interpret the data that we find from our experiments. So we are in the process of producing dopaminergic neurons, which are the key cells in this particular disease. The goal is to demonstrate a differentiated disease phenotype. In Parkinson's, it is lower levels of dopamine that are being secreted, and develop assays that we can use for screening compounds that could rescue this disease phenotype. And that's the end of my talk. Thank you.
Are you having any other questions that we can answer? I mean, I've seen some of you have already left because we are running a little over, but if there are really urgent questions, I think the speakers would be happy to stay and answer them. Yeah?
How long will it be before you can go from the in vitro trials to the in vivo trials to validate the methodology that your company's developed?
Our plan is to be able to be in the clinic in 3 years. So what is very possible is that we will not even worry about animal models, because the animal models have not recapitulated really the disease. And we hope to convince the FDA that the in vitro clinical trials, starting already with patients, would be something. So we'll use animals mostly for toxicity, and try to push into the clinic.
I was wondering, is there any other websites or whatever, any materials I can bring back?
So again, CERM has, the California Institute of Regenerative Medicine has a wonderful site with I actually, when I started to teach stem cell at San Jose State, I used this site a lot. They encourage scientists and teacher educators to use this site. Beautiful pictures, a lot of videos for the most popular scientists that work on the field of stem cell. I very strongly encourage you to check their website. Very valuable, a lot of very good information in there.
Yes. So you may have heard when I mentioned that we are using retroviruses to introduce the 3 or 4 factors. This means that we are using integrating means, genome integrating means. So this already creates a cell that is not pristine. So that's just not even thinking about the problems of creating iPS cells for different people, characterizing them, producing large levels. Not only that, if we are thinking of differentiating these cells to the type of cell of interest, you have to make sure that 100% of the cells become that type of cell, and that you don't even have one cell in the body that's less differentiated and can become a cancer cell. So the field is simply not ready for that.
So you're not saying just in your particular disease, but overall it's just not ready?
Yes, the field is not ready yet. It's a very young technology. It started just 4 years ago with Shinya Yamanaka from Japan. And it caught up like fire, of course, because of its power. But we are not ready for that as yet. It's too young of a technology.
Well, I'd like to thank you all for joining us and thank our speakers for a really enlightening discussion. As I said before, this is an evolving field that's growing so quickly, it's almost hard to keep track of what's going on. And again, thank you so much for coming, and we hope this has been an enlightening afternoon for you all. Thank you.